Welcome back to the channel. Here in this video, we are going to take a comprehensive look at the prokaryotic plasma membrane, also known as the cytoplasmic membrane. This thin structure composed of lipids surrounds the cytoplasm and defines the boundary of the cell. We will explore its intricate structure, the fascinating chemistry that makes it so effective, and the various functions it performs to keep prokaryotic cells alive. And in the last part of this video, I will discuss how the plasma membrane of pathogenic bacteria can be used as an excellent drug target for disease treatment. Not only that, the lipid composition of the plasma membrane for a certain bacteria is unique and can be used to identify a particular microbes in microbiology lab. So let's start by understanding what the cytoplasmic membrane is. Imagine a balloon, it's just like a cell, and the skin is like the plasma membrane, separating the inner compartment from the external environment. Unlike balloon, the plasma membrane is fluid in nature. It's only about 6 to 8 nanometers wide, so tiny that it can only be observed under an electron microscope. When viewed under the microscope, it appears as two dark lines with a lighter area between them, and this structure is known as the unit membrane. But why is this membrane so crucial? Simply put, if the membrane is damaged or broken, the cell loses its integrity, and the cytoplasm, the gel-like substance inside the cell, leaks out, and the cell ultimately dies. This makes the cytoplasmic membrane essential not just for protecting the cell's content, but also for ensuring its survival. To understand the complexity of the cytoplasmic membrane, let's discuss the most widely accepted model for its structure, the fluid mosaic model. It is proposed by Singer and Nicholson. This model gives us a detailed understanding of how the membrane's components are organized and how they function. According to this model, the membrane is a lipid bilayer, meaning it consists of two layers of lipid molecules. It is fluid in nature, not rigid like balloon. Within this bilayer, proteins are embedded, much like boats floating in a lake. The term fluid mosaic comes from the idea that these proteins and lipids are not static. Instead, they move freely within the membrane, creating a dynamic and flexible structure. The backbone of the membrane is made up of phospholipid molecules, which are arranged in two parallel rows. In phospholipids, a glycerol molecule is present, and its carbon atom number 1 and 2 are joined to two fatty acids via ester bonds. A phosphate group, which is negatively charged, is attached to the third carbon of glycerol. The glycerol adopts a structure called syn. As a result, both fatty acids are in this side and glycerol and phosphate group are in this side. The hydrophobic tail, made up of fatty acids, point inward, away from the water, creating a hydrophobic core. Meanwhile, the hydrophilic head, composed of glycerol and phosphate, face outward toward the water, either inside the cell or in the external environment. When the fatty acids are straight, they pack tightly, making the membrane rigid and impermeable to most molecules. To maintain the fluidity, some unsaturated fatty acids, which are not tightly packed, are incorporated, making the membrane more fluid. By carefully selecting the appropriate fatty acids, different bacteria can maintain the fluidity of the plasma membrane at various temperatures. As you can see, if the chain length increases, the melting point increases, and the melting point decreases if the degree of unsaturation increases. Bacteria growing at lower temperatures will incorporate fatty acids with lower melting points into the membrane phospholipids, ensuring that the membrane remains fluid and functional even in cold conditions. Next, let's talk about the protein embedded in the membrane. These proteins can be broadly classified into two types: peripheral and integral proteins. Peripheral proteins make up about 20 to 30% of the total membrane protein. They are loosely attached to the surface of the membrane, meaning they can be easily removed because they are soluble in aqueous solutions, and these proteins play a crucial roles in various cell processes, including signaling and maintaining the cell shape. Integral proteins make up 70 to 80% of the membrane proteins. They are deeply embedded within the lipid bilayer. They are not easily removed and are insoluble in aqueous solutions once separated from the lipid membrane. Integral proteins are involved in a variety of essential functions. such as transporting molecules across the membrane, acting as an enzymes, and participating in cell communications. Moving on, let's discuss the carbohydrates associated with the membrane. Many of the proteins and some of the lipids of the outer surface of the plasma membrane have carbohydrates attached to them. When proteins are attached to carbohydrates, they are called glycoproteins, and when lipids are attached to carbohydrates, they are called glycolipids. These carbohydrate structures are not just for decoration. They serve several important functions. The glycoproteins and glycolipids help to protect the cells, lubricate it, and are involved in cell-to-cell interactions. For instance, they play a key role in the immune system by helping cells recognize each other, distinguishing between the body's own cells and foreign invaders. You can see in this picture that the length and the sequence of the oligosaccharide chains are different, and this imparts antigenicity. Now, let's explore how prokaryotic membrane differ from the eukaryotic cell. One of the key differences lies in the presence of molecules that strengthen the membrane. In some bacteria, the cytoplasmic membrane is strengthened by molecules known as hopanoids. These somewhat rigid planar molecules are structurally analogues of sterol, which strengthen the membranes of eukaryotic cell. Interestingly, many eukaryotic cells that lack a cell wall rely on sterols for membrane integrity, whereas bacteria use hopanoids for similar purpose. Thus, hopanoid synthesis can be a potential drug target for treating bacterial infection. While we have discussed the general structure of prokaryotic membrane, it is important to note that there is a significant difference between the membranes of bacteria and archaea, the two major groups of prokaryote. Archaea, often known for inhabiting extreme environments, have membranes that differ fundamentally from those of bacteria and eukaryotes. One of the key differences is the type of linkage that connect the lipid molecules. In archaeal membrane, the hydrocarbons are attached to glycerol by ether linkages, as opposed to the ester linkages found in bacteria and eukaryotes. Ether linkage is more stable, particularly at high temperatures, making them ideal for organisms living in extreme heat. The fatty acid chain in archaeal membrane are often branched, which influence the fluidity of the membrane at high temperature. This branching helps to maintain the membrane integrity and function in harsh conditions. In some archaea, the two glycerol groups are linked to form extremely long tetraether lipids. These lipids can span the entire membrane, creating a monolayer instead of a bilayer. Lipid monolayer are particularly resistant to heat denaturation, making them a common features in hyperthermophiles, organisms that are found at temperature above 80 degrees centigrade. With an understanding of the structure and chemistry of the cytoplasmic membrane, let's now explore its various functions. The membrane is not just a selective barrier. It plays an active role in maintaining the cell's health and facilitating its functions. One of the primary functions of the cytoplasmic membrane is to act as a selective barrier. It prevents unwanted substance from entering the cell while allowing essential nutrients to pass through. Similarly, it regulates the export of waste products out of the cell, ensuring that the internal environment remains stable and friendly. It is involved in the generation and use of proton motive force, a process essential for ATP production, the energy currency of the cell. By maintaining a gradient of protons across the membrane, the cell can produce energy to drive various biochemical reactions. Finally, the cytoplasmic membrane serves as an anchor for a wide range of proteins, and these proteins are involved in transport, helping to move molecules in and out of the cell, as well as in bioenergetics and chemotaxis. Chemotaxis, in particular, is the process by which the cells move towards or away from the chemical stimuli, a critical function for survival in changing environments. Now, we will look at the antibiotics that specifically target the plasma membrane of bacteria. Daptomycin is a cyclic lipopeptide antibiotic that is used to treat drug-resistant gram-positive bacteria. In the presence of calcium, daptomycin molecules typically oligomerize, creating a pore in the membrane, and this pore allows essential ions to leak out, that ultimately leading to the death of the bacteria. Another antibiotic, polymyxin, are surface-active cationic detergents with bactericidal properties. They work by interacting with the phospholipids in bacterial cell membranes, disrupting their structures, and these antibiotics are a group of N-monoacetylated decapeptides. Polymyxin B and polymyxin E are used clinically to treat infection caused by gram-negative bacteria. However, due to their neurotoxic effects, they are typically used externally. The identification of pathogenic bacteria from patient sample is one of the major tasks in microbiology labs. For the identification of bacteria, nowadays the membrane lipid composition is also analyzed by gas chromatography or using mass spectroscopy. The chromatogram or mass spectra are then matched with the reference data to identify a particular microbes in patient samples. Here you can see that different bacteria shows different spectra. If the spectral analysis of an unknown bacteria match, we can easily identify the microbes. So that's all for now. If you found this video informative, be sure to like, share and subscribe to my channel. I have plenty more educational content coming your way. So stay tuned and thanks for watching.